When you hear the word cancer, time seems to stop. But almost immediately, a new clock starts ticking. You are suddenly waiting on tests, waiting on specialists, and waiting to find out what happens next. Traditionally, what happens next is a protocol known as the standard of care. Oncologists prescribe a baseline treatment, and then you wait. It can take 8 to 12 weeks of grueling chemotherapy or radiation just to see if the tumor shrinks. If it doesn't, doctors have to pivot to a second-line option and start the clock all over again. It is an exhausting process of trial and error. Today, there are more than 300 FDA-approved cancer treatments, interacting with a practically infinite number of unique genomic mutations. No single human brain can recall and cross-reference every relevant data point and clinical trial available at any given time. Applying a population average treatment to an individual's unique genetic mutation lacks the necessary precision. It's the clinical equivalent of trying to shatter a target with a heavy sledgehammer when the situation requires the hyper-focused cut of a surgical laser. The modern oncology pharmacy is robust, but its efficacy is trapped behind a predictive barrier. The current system relies on observing a patient's reaction to a drug only after they have already endured the physical toxicity of a potential failure. To break that barrier, artificial intelligence acts as a medical detective. These systems instantly aggregate and contextualize decades of global clinical trials, papers, and genomic data that would take researchers lifetimes to process manually. This chart shows the historical trade-off between a model's prediction accuracy and its explainability. In medicine, black box algorithms are an ethical liability. Doctors cannot rely on life or death recommendations without seeing the logic behind them, which drives the move toward explainable AI. This plot maps the trajectory toward what researchers call strong AI. These models aim to achieve the high learning performance seen at the top of the chart while maintaining the complete transparency required for clinical validation. This diagram shows XAI systems using nanodatasets, physical, chemical, and biological data, to build a digital twin of a patient's tumor. The algorithm runs virtual experiments on this twin to optimize dosing and achieve targeted delivery. Oncology transitions from a series of physical trials on a human subject into a calculation performed in a digital environment. Naturally, your immune system uses T cells to defend the body, but these cells often struggle to identify and bind to cancer. Tumors are highly varied and adept at masking themselves, making it difficult for the immune system to recognize the threat. Researchers at the Technical University of Denmark are using AI to solve this recognition problem. Their platform designs custom protein-based binders that physically dock with a patient's T cells, providing specific instructions to identify and eliminate cancerous cells with extreme precision. Traditionally, discovering personalized molecular binders takes years. AI compresses this timeline to just 4 to 6 weeks. Without sacrificing safety, the algorithm screens out toxic molecules. Only the safest data points pass through the filter to become a physical drug. In a race against a mutating disease, these algorithms are providing patients with the one resource they cannot manufacture: time. There is a practical requirement for this level of precision. To perform the initial genomic sequencing and drug sensitivity tests, these AI models require access to high-quality living tumor cells from the patient. This requirement has led to a tissue preservation crisis. Standard hospital biopsies often exhaust the available sample on basic tests, or the tissue is degraded and discarded. Once that biological material is gone, the patient is permanently locked out of these AI-driven treatment options. This makes tissue cryopreservation an urgent priority. Demanding that a biopsy sample be preserved immediately following the procedure ensures that a patient's biological data remains available for future AI modeling and clinical trial matching. Patients who achieve significantly better outcomes are not necessarily using secret medications. They are using their biological data to be matched to the correct existing drug from day one, avoiding the dead ends of traditional trial and error. By automating the processing of massive datasets, these tools allow oncologists to focus on the human side of care. We are moving toward a future where a cancer diagnosis is managed as a treatable condition rather than a terminal countdown.